Welcome today to today's micro seminar for this week. We're lucky to have a speaker, Dr. David Zeevi, who is a fellow at the Rockefeller University, and he's going to be talking about some ocean microbes, I think, and some understanding the genetic code within ocean microbes. So David started out as an undergraduate at the University of Tel Aviv with a B degree in computer sciences and life sciences. Did a master's at University of Tel Aviv University with Tal Pupko, and then completed his PhD in systems biology at the Weizmann Institute with Aaron Siegel. So I would, just want to say thank you. Thank you for joining us to present your science today, David, and I'll hand it off to you. So I will see you at the back end. Any questions I'll take through Twitter. If you have them there, post them. And oh, sorry, my computer's doing weird things. So I will take any questions through Twitter, and then I will ask them at the end. So if you have questions, please post them there. If the comments on YouTube are open, feel free to post the questions on YouTube as well. But otherwise, take it away, David.
All right, thank you, Dave. Thanks so much for the opportunity to speak at this micro seminar. I've been following this YouTube channel and enjoying it very much. So I'm going to talk about a recent work that's about to be published soon, I hope. And this work was done in tandem with Liat Shenhav, formerly from UCLA. And more recently, she moved into Rockefeller University, and she's at the office across the hall. And the thing I like the most about this project is that we started with a very, very clear motivation, but like often happens with scientific projects, we went through some very interesting twists and turns and into rabbit holes and out of them again. And we got to somewhere completely different, which was interesting and surprising for us. So I'm going to start with a very clear motivation, and I'm going to take you through the twists and turns. And hopefully, in the end, you'll see that I'm not really going to pull you out of the rabbit hole. We're going to just stay there. So our motivation was marine pollution. And this is a growing problem around the world. And the reason we looked into marine pollution is that even though your elected officials or government or whatever doesn't really care about marine pollution, we thought that perhaps microbes do care about marine pollution. And they're developing interesting mechanisms to deal with this pollution or to avoid it. And we wanted to look into marine microbiomes to find new genes that microbes use to avoid or to combat pollution. So in order to do that, we put together microbiome data from about 700 deeply sequenced samples from around the world. Some were made available through the Terra Oceans Initiative. Some were made available through Penny Chisholm's lab. And using these metagenomic samples, we calculated evolutionary conservation metrics. At the same time, we collected data on pollution sources from many different papers and many different data sources that were around. So what we're seeing here are data on plastic particles in the ocean, data on shipping routes. We have oil rigs around the world, power station. Not shown here, we have organic and inorganic fertilizers, heavy metals, and several more. And this was a big effort. You see these nice maps, but actually harvesting the data and using it took a lot of time and effort. And what we want to study, what we want to do is basically to take the marine microbiome samples, calculate sequence conservation metrics, I'm going to get into it soon, take the marine pollution data, and get the value of this specific pollutant wherever these samples were taken, and then cross-correlate them or associate these two metrics, the conservation metrics and the pollution metrics, and find genes that are only conserved in regions where there is pollution. And our thought or our notion was that these genes would be good candidates for treating these pollutants either in a bioreactor or maybe even in situ. Right? It would definitely teach us more about how microbes react to these pollutants. So we went about to calculate sequence conservation metrics. And I'm going to take a short detour and explain how we do that. So basically, DNA is transcribed into mRNA, and mRNA is translated into protein. And each codon triplet in the mRNA is translated to an amino acid. But since there are 64 codon triplets, or 61 if you exclude stop codons, and only 20 amino acids, some amino acids are coded for by more than one codon. So you can see in the table here, which is the standard genetic code, that proline is coded for by 4 codons, histidine by 2, and arginine by 6. There's this, these 4 and these additional 2. And basically, we took these ocean samples, we calculated single nucleotide polymorphisms, and we went about to calculate purifying selection by comparing the rate of non-synonymous mutations, which are mutations that change the amino acid, and comparing them to synonymous mutations, which are mutations that do not change the amino acid context. And basically, if this ratio is close to 0, it means that for that specific gene or for that specific genomic region, mutations that are changing the protein sequence are not tolerated or hardly tolerated, and this is called purifying selection. And basically what we were looking for when we were looking for this, these genes that are conserved, we were looking for things that have PNPS close to 0 only in regions where there is pollution. But what we found instead was that everything in the marine microbiome, or almost everything, had PNPS ratios very, very close to 0, or very, very low PNPS. So it seemed to us that everything was under very strong purifying selection. And this was true even when comparing the marine microbiome samples that we had to human microbiome samples from 2013 Nature study. And basically, that surprised us. And we wanted to understand, why is there such strong purifying selection across marine microbes? And this is the first rabbit hole that we went into. So in order to understand what could lead to this type of selection, we looked at the immediate suspects, which are usually the environment these microbes are in. So we used environmental measurements that were taken alongside the microbiome samples. And what we found is that these PNPS ratios, this conservation metric, is highly associated or highly correlated with environmental measurements. So what we're seeing here is our histograms of correlations. And coming from the world of human microbiome, I hardly ever saw correlations as high as we've seen here. So we're seeing predominantly positive correlations between PNPS and nitrate, predominantly positive correlations between PNPS and depth, and negative correlations between PNPS and oxygen, meaning that in low depths, shallow depths, and in low nitrate concentrations, there is low PNPS, meaning higher conservation. But all of these metrics are associated with one another. So we wanted to account for this correlation structure, but still measure the effect that the environment has on PNPS. So we modeled it using a linear mixed model. And I'm not going to go on into all the gory details of the linear mixed model, but we saw that the variance explained by the environment in PNPS was very, very high. And when we compared it to the same data shuffled, we saw that it was very significant as well. So this, again, surprised us. Why does the environment affect PNPS so much? So we went into the literature and found that there were cases shown where resource conservation affected nucleotide sequences and protein sequences of microbes. Mainly what we saw is that when there's a lack of nutrients, there is a shift into not incorporating these nutrients into DNA and protein sequences. And just to illustrate what I'm saying is, I'm going to show you 2 microbes. One, the blue one, A, is a wild-type microbe, and B is a microbe with a point mutation, a T2 gene mutation, in a certain gene. This mutation in and of itself causes an increase in one nitrogen atom because a CG pair has one more nitrogen atom as compared to an AT pair. But when it's translated into protein, it causes a leucine to arginine mutation, which costs the microbe 3 additional nitrogen atoms per copy of the protein. Now, if these microbes are in a nitrogen-abundant environment, they would be similar because they could get enough nitrogen to go around, and the mutation may fix if it's not deleterious to the gene. However, if nitrogen is limited, B is going to take longer, the red one is going to take longer to grow because it's going to take longer to get all the nitrogen it needs to grow, especially if these genes are highly expressed. And then B is going to be disadvantaged, and the environment over a lot of generations is going to favor A. And basically, looking at this, we saw, well, this could explain perhaps what we're seeing in terms of conservation against incorporating nutrients into protein and DNA sequences. And one important understanding that we had was that if the expression of these genes is higher, the higher the expression of the genes is, the higher or the stronger the selection is going to be, because if we have a higher expression protein, then each of these mutations is going to translate to potentially thousands or even millions of nitrogen atoms that have to be incorporated in each cycle or in each replication of this microbe. And indeed, when we compared genes that had high expression to genes that had low expression from a different data set altogether, we found that the variance explained in highly expressed genes is much higher, and very significantly so, than the variance explained in lowly expressed genes. The variance explained by the environment, of course. We wanted to look into a different set of genes altogether, and we looked at secreted genes. Now, in these genes, it doesn't really matter what, how many proteins are, how many nitrogens are put into what the expression of these genes is. Any resource that's being put into these genes is thrown away from the cell. And indeed, in these genes, the variance explained by the environment is significantly higher than all other genes. And this is where we get into the second and more deeper rabbit hole. We wanted to see how universal this selection is. And this is where it gets really cool. Specifically, we wanted to see whether this resource conservation was made easier by the amino acid choices of the microbe and the structure of the genetic code. This is the structure of the genetic code. And many studies have been made on the optimality of the genetic code and found that the code is structured to minimize errors. And most prominently, minimizing errors that change hydrophobic amino acids to hydrophilic amino acids and vice versa. But what we asked is, does it always also minimize adding nitrogen and carbon given a random mutation? And in the case of nitrogen, the reason that we had this intuition is because of this neat square of nitrogen-rich amino acids. And what this neat square means is that if you're, for example, valine or alanine, which are very, very common amino acids, you can't become nitrogen-rich just given a single mutation. So you have to have a mutation in the first position and then a mutation in the second position, or vice versa, in order to become nitrogen-rich. And this looked to us as sort of a protective mechanisms against becoming nitrogen-rich by mistake. And when we looked at carbon and oxygen in the genetic code, we saw that maybe this also happens with carbon because there's sort of a gradient towards the top of the table in terms of the number of carbon atoms. In oxygen, we didn't see this type of selection or this type of organization, but we wanted to check that as well. And the way we quantified the cost of random mutation was by looking at the expectation of the number of nitrogen atoms that a single mutation would add. So we call that the expected random mutation cost. And I'm going to walk you through this calculation because if we're comparing the genetic code to some other random genetic code, then we have to know what we compare it on. And this expected random mutation cost is basically the probability of starting at some codon, let's say threonine, times the probability of mutating to a different codon, let's say arginine, times the cost of transitioning from one amino acid to the other amino acids in terms of added number of elements, in this case, nitrogen. And we do that for all pairs of amino acids. So basically, if I'll walk you through this calculation again, we start with ACA coding for threonine, we mutate to AGA coding for arginine. So we estimate the probability of starting at ACA using the average relative abundance of threonine codons, the probability of mutating from ACA to AGA using the probability of mutation from C to G in our data set, and the number of added nitrogen atoms is 3 in this case. So we multiply that, and we do that for all pairs of all pairs of codons. And then what we can do is to shuffle the genetic code and create a null distribution. What we're doing is creating 1 million of these genetic code options or these hypothetical genetic codes, and then we're comparing them to the standard genetic code in terms of nutrient conservation. And what we find is that the standard genetic code is more conservative given a random mutation than all but 142 codes in terms of nitrogen conservation, and all but 119 codes in terms of carbon conservation. And when we demand that the shuffled genetic code would be better than the standard genetic code in conserving both nitrogen and carbon, we found that only about 1 out of 1,000 genetic codes does better than the standard genetic code in conserving both nitrogen and carbon. Now, this was done with the abundances of codons that were calculated from marine microbes, and we wanted to see how it extends to abundances of codons from the entire from all domains of life. So we recalculated that with codon abundances from across 5 kingdoms. And since we did not know the mutation rates from one place to the other in the genetic code, we calculated it with a range of transition transversion rates. And what we find is that for all organisms tested and for all transition transversion rates tested, there is a significant optimization for nitrogen and carbon combined in the standard genetic code. This is showing the human genome. So now we wanted to ask, how does this optimization compare to known optimization mechanisms? And basically what we're looking for is how it compares to optimization mechanisms maintaining the polar requirement of amino acids and hydropathy of amino acids. These are metrics that scientists came up several decades ago to measure how the genetic code is optimized in terms of hydropathy or polar requirement. And what we're seeing here is in green, the hydrophobic amino acids, and in brown, the hydrophilic amino acids. So you can see that it's, in order to mutate from hydrophobic to hydrophilic amino acids, you have to have a mutation in the second position of the specific codon. And we wanted to see how this optimization compares to what we found. So we reran our calculation with these metrics and found that the known optimizations for structure and function of the genetic code are on the same scale as the ones for nutrient conservation. We then asked if there's some sort of overlap between these metrics and the metrics that we found on nutrient conservation. So, for example, it could be the case that if you optimize for hydropathy, you don't need to re-optimize for nitrogen and carbon because these two mechanisms overlap. So we looked at a bunch of random genetic codes that were better than the standard code in maintaining hydropathy. If there was a big overlap between hydropathy and, for example, nitrogen or carbon, we expect that these genetic codes that are better than the standard genetic code in terms of hydropathy to also be better than the standard genetic code in terms of nitrogen or carbon. But this is not the case. When we're looking at the tail of the distribution here, that's better than the standard genetic code in terms of hydropathy, we still see optimization in terms of nitrogen and carbon. So basically, we have two additional mechanisms of optimization in the genetic code that weren't known so far, and they extend to all domains of life. So next, we asked, are the structural characteristics, namely the nitrogen square that we saw, making the code more parsimonious in terms of nitrogen conservation? So basically, does this structure of nitrogen amino acids organized in a square, topologically a square, is better than other organizations? So first of all, as a thought experiment, we tried to think about what would be the worst possible organization of nitrogen amino acids. So I'm going to give you a moment while I take a sip of water to think about what would be the worst organization of nitrogen amino acids. Anyway, what we came up with is a diagonal. And this would be really bad because unlike with a square where some amino acids or some codons can avoid becoming nitrogen rich because they take only 2 mutations to become nitrogen rich, when there's a diagonal, from every point in the genetic code, you can become nitrogen rich not in one way, but in 2 different ways. And indeed, when we compared all square arrangements, and this is also, for example, a square arrangement, it's a topological square, to all diagonal arrangements, we see that square arrangements are more conservative in nutrients as compared to diagonal arrangements and all other arrangements. Finally, in these cases, we mutated the first and second position of the genetic code, and we wanted to see if there is some effect on the third position as well. And in order to look at that, we looked at all of the codons that start with adenine, with A. And the reason that we looked into that row, that specific row of the genetic code, is because when we examine threonine codons, for example, we see that there's a clear difference between the top 2 codons and the bottom 2 codons. Because if you mutate the top 2 codons, you end up with a C2G mutation in the second position. You end up with serine, which has low carbon and low nitrogen. But if you mutate the same mutation with the bottom 2 codons, the ACA and ACG codons, you end up with arginine, which has high nitrogen and high carbon. And in fact, what we're seeing is that taking into consideration differences between GC content in genomes, ACT is much more popular than ACA, and ACC is much more popular than ACG. And we see that this extends as well to isoleucine codons. So to summarize this work, basically, I showed you that nutrient limitation is a strong selective force in the ocean and probably in other environments as well. I showed you that the genetic code facilitates conservation of carbon and nitrogen, and this is orthogonal or added to known optimization mechanisms of the genetic code, and that some structural characteristics of the genetic code make it more streamlined in terms of nitrogen and probably also carbon conservation. The last thing I want to talk about is that there's no apparent oxygen optimization in the genetic code. We saw optimization for nitrogen and for carbon, but not for oxygen. And it got us thinking that since the genetic code is common to all of life on Earth, basically maybe with just minor changes, it may have been there for the last universal common ancestor. And that means that it may reflect the conditions that were around for in the beginning of life. So a scarcity of carbon and of nitrogen may have molded the genetic code to what it is now, and perhaps oxygen wasn't a problem or wasn't even around to be considered as a problem or as something to optimize for. So finally, I want to grab you by the legs and pull you out of the rabbit hole and talk about pollution again. Now that we know that that there's an underlying layer of selection for nutrients, we can account for it when we're detecting pollution based on selection metrics in two ways. We can computationally remove just look at the residuals of selection after we accounted for this resource-driven component of the selection, or we can even look at the genetic code and look for the regions in the genetic code, such as valine and alanine, that are under weak resource-driven selection, but strong selection for protein structure and function. And with this, I'd like to end, and I'd like to thank Liat and Hav, who I showed you in the beginning, and also tell you that Rockefeller University is hiring a fellow for next September, and I really recommend this position. Thank you.
All right, awesome. I actually hadn't read the preprint before, so it was an awesome view into that work, and it's kind of really cool stuff. So I'm going to cruise. So if anybody has questions, again, please feel free to post them to Twitter and at MicroSeminar, and I'll catch them there. But to start off, I do have a couple of questions that just popped up in my own head that I'm wondering if you've looked into. And they're related, but different. And my kids are running around very loud in the background, so apologies for that. So in terms of microbial communities in the ocean, I know there's a lot of talk about streamlining of genomic content. You know, some strains lose a bunch of genes, some strains don't. So have you looked at parsing out big genomes versus streamlined genomes in terms of purifying selection? Like, are they the same or are they different? Like, is there something there in terms of more resource optimization in the streamlined ones versus the bigger ones, basically?
So we didn't look at it per se. We just validated our results that we looked at metagenomics and we wanted to validate it on in other ways. So we did validate it with single cell genomes of SAR11, SAR86, and Prochlorococcus. But we didn't check whether there's more streamlining or less streamlining in big genomes in terms of nitrogen conservation. But what I'm thinking is that it's probably affected by resources, but also by the effective population size. When there's more competition, you want to be as stringent as possible or as parsimonious as possible in the number of atoms that you take up from the environment, because taking up less than your neighbor makes you more fit, in a sense.
And then, so following on, and I'm guessing that you haven't looked at this level either, but I know that there's a lot of phage in the ocean. And so were you able to parse out phage genomes versus bacterial genomes? Because I can imagine there might be different resource allocations in terms of things that need other organisms to grow versus kind of autonomous dividing ones.
Yeah, that's a really interesting perspective, because phages shouldn't care, right, about the microbe conserving resources or not. Well, maybe they do, because if there's fewer resources, then you can make fewer phage genomes. But we haven't looked at it. It would be interesting too.
All right, looks like we got a question from Twitter. So the new model of resource-driven selection of the genetic code is compelling. Does it refute other previous explanatory models of the conserved code?
No, not at all. It's just orthogonal to other models. The other models are still explanatory of the genetic code, but if we once thought that the genetic code was only optimized for structure and function conservation of the protein, now we have an additional layer of conserving also resources.
Have you been able to look at outside the marine environment where the resource utilization? I know that there's a lot of weird genetic codes out there and some protists and stuff like that. So have you been able to kind of start looking at the other kind of weird genetic codes to see if there's anything there in terms of resource allocation, or is that kind of outside the realm of stuff?
Well, the weird genetic codes aren't that weird. That's the thing. They're just a tiny bit different than the standard genetic code. So the standard genetic code is really mainly quite frozen. And this is probably because it's really hard to change. Imagine that if you want to change the genetic code, you have to change both tRNAs. You mean, the genetic code is not written anywhere in the cell. It's just the specific tRNAs with their anticodons. You have to change these, and you also have to change all of the protein coding sequences in the genome. So going around and changing the genetic code is very hard. And this is why it nearly only happens in genomes that are very degenerate, like mitochondrial genomes that don't have a lot of genes. So they can more easily mold them or change the genetic code a little bit. And even then, it's not a big change usually.
Cool. I'm going to take one last cruise around Twitter and see if there are any questions. Doesn't look like it. So I want to thank you again for an awesome micro seminar. And I also, for anybody out there, I tweeted out a link to the preprint that's in Press at Science. And so if this work was compelling, take a look at the preprint, because you'll be able to kind of go through a bunch of this as well in print form. So again, David, thank you for sharing this all with us. And I look forward to seeing this out in print for real publication soon. So
thank you.